Hello, my name is Russell Garlick. I'm the Chief Scientific Officer for LGC Clinical Diagnostics. Thank you for joining today's presentation. I'll be talking about the challenges of cell-free circulating tumor DNA NGS assay validation. I'll review the importance of quality control materials, go through analytical validation protocols towards standardization, talk about the technology we use to develop and manufacture fit-for-purpose ctDNA reference materials, talk about the R&D pipeline, and we don't have a live Q&A session, but please feel free to submit any questions you might have to me at my email below here, russell.garlick@lgcgroup.com. I'd be happy to answer your questions. So oncology-based cell-free DNA assays, there's really three reasons we're advancing this technology so quickly to patient management. First is cancer detection. Is the patient infected? Is the patient with cancer or without cancer? Secondly, tumor profile is so important. What mutations are present, and knowing those mutations, what treatments are available. Hence, there's been a lot of work using ctDNA assays as companion diagnostics. It's advancing quite nicely. And we've moved into the whole era of immuno-oncology. Wonderful treatments to actually cure cancers are underway. And tumor mutational burden, both solid tumor and blood, is now being used to identify those patients who are going to respond to immunotherapy. And lastly, monitoring. Is the treatment working? Does the cancer come back? And if it comes back in a metastatic mode, can you detect those variants in the blood? I like to refer to that as measurable residual disease. So lots of reasons cell-free DNA towards oncology is being applied in the clinical laboratories. So what's the importance of QC material? I'd like to take these highlighted points from Dr. Greg Zongales, who presented this information at the AMP meeting in 2018. First, the purpose of QC materials can identify possible variations that may compromise assay integrity and compromise the confidence in the test results. Classic QC use of these materials. Ensures the accuracy and reproducibility of lab test results used in patient care. QC materials should be very similar to patient specimens so they can monitor the entire workflow from extraction, library prep, sequencing, and bioinformatics reporting. Commercial materials can provide a consistent supply for long-term use. With ctDNA, there's always a sparse number of samples available. We can manufacture materials that can ensure this consistent supply in long-term use for QC. And classically, we want to make sure QC is done each time a patient sample is run. And with good QC materials that you know and expect the results, it's ideal to use these materials to troubleshoot complex instrumentation issues that you may have. So ctDNA testing by NGS. We've really done a wonderful job advancing this technology to routine patient diagnostics. We've moved from research phase through clinical to laboratory developed tests, and now we're in the phase of companion diagnostics, identifying mutations with an assay for specific patient treatments. And there are many approaches now being taken for IVD approval. The clinical practice guidelines recognize the clinical utility of ctDNA assays, particularly with non-small cell lung cancer and EGFR tyrosine kinase inhibitors, selecting the right therapies for patients who have those mutations, and also being used for persistence in post-resection using ctDNA. Now, each regulatory milestone we've advanced to requires more pre-analytical and analytical validation, increased stringency, which means increased investments for the laboratories using these tests and the manufacturers of the products. And fit-for-purpose reference materials help advance and reduce the cost of the overall process. Multiplex reference materials are designed and aligned for next generation sequencing where we're interrogating multiple cancer genes and can help accelerate approvals. These are manufactured products, they're patient-like, and they can address the limitations we have with patient samples in the laboratory, both number and amount available per patient. Typically, a drawer is one tube will end up with 6-7 mils of plasma. That is not enough for continuous validation and QC over a long period of time. And furthermore, these manufactured materials can include many types of variants and classes of variants that cause cancer. So analytical validation protocols. These can address the assay challenges. ctDNA is shed from apoptotic cells or necrotic cells at very low abundances, down to 0.1% to 5%, a little fraction. The other challenge we have is that with NGS assays, you're interrogating multiple genes, but you're also interrogating multiple types of fusions. SNVs, insertions, deletions, copy number gains, and fusions all require validation. These are complex workflows from drawing the blood to the extraction, ensuring that's consistent and not variable, issues around stability, issues around sensitivity. So there's lots of challenges validating these assay protocols. We've worked extensively with organizations like BloodPack addressing some of these challenges. And we're really at the phase of development where standardization will help accelerate adoption, overcome caution adopting this new technology. It really establishes legitimacy of the test results across the labs and the platforms. So BloodPack is a multi-faceted organization with members from regulatory agencies like FDA, clinical laboratories, IVD manufacturers, next generation sequencing manufacturers, and organized to help standardize and advance NGS testing. And within BloodPack, the BloodPack Analytical Variables Working Group, led by Jim Godseed at Illumina, just published in Clinical Chemistry a fantastic product on the generic protocols to ensure robustness and standardization. And I want to speak to some of the recommendations made by this BloodPack group next. So here's a table that summarizes the protocols intended for validation of these NGS assays. This column is the protocol, the approach, and the types of samples that a laboratory can consider to validate that particular part of the protocol. So limited blank, you should be using negative patient samples. Analytical accuracy, of course you have to include your negative samples as well, but you also need 10 to 20 patient samples for each variant. Again, this is the recommendation from BloodPack published in Clinical Chemistry. For linearity, you need to work across the working range. And here you can use a combination of contrived samples, manufactured samples that are patient-like, or pooled patient samples. Limit of quantitation, where you need to do this for all variant types. In replication, and you need to be able to dilute in wild-type ctDNA course test and use different reagent lots. And again, this is a large part of the study. So using contrived materials is recommended, or pooled patient samples. For repro repeatability studies, you need to use 3 manufactured lots, follow the CLSI protocol, 3 sites, 2 operators, qualitative and quantitative study, lots of statistical norms. There's lots of samples required for this. Again, clinical samples are always preferred, but the recommendation is you can use contrived manufacturing samples to supplement. Looking at interfering samples, particularly around extraction and detection, you need to test your worst case levels. And again, contrived, pooled, or normal. Guard banding, very critical for pre-analytical and analytical. Again, contrived or pooled. And then specimen stability. Here, of course, you can only use real patient samples. And again, the contrived samples are really key to be able to do true validation of these NGS assays over time. So contrived sample can be used to analyze regularly to detect possible sources of errors and monitor assay performance. And it can reflect the size distribution of your samples, full process controls, extraction through detection. It can be formulated at the right levels relative for your assay. So let me say some more about contrived materials and how we at LGC develop and manufacture these products. So there's really 4 different ways you can think of manufacturing contrived materials to support ctDNA NGS assays. We want to make them as patient-like as possible. And then, as I mentioned earlier, you can do pooled patient samples. But the issue with pooled patient samples is you can never be sure exactly what is in there since you're pooling samples. You just know what you can actually detect. So on the contrived samples, there's really 4 approaches. Fragmenting cell lines. Unfortunately, there's a limited selection, and most do not have SNP matched normals to pass contamination checks. Fragmented engineered cell lines, an issue with cost around customization, which we need to do when you consider all the types of variants you need to look at. Aneuploidy makes obtaining defined VAS very difficult. Cell culture supernatants have been used, growing these cells and then harvesting the cell culture soups. Again, very costly as far as customization, very low yields. Aneuploidies make it difficult to define, to obtain these variant little fractions, and very low yields. Our approach at LGC is to use the SeraCare branded fragmented biosynthetic spike-in to a genome in the bottle background standard. So and these are designed, they are multiplexed, they can be adjusted to whatever little fraction you want or content, whether it's SNVs, insertions, deletions, etc. And we make them very patient-like, and I'll say some more about that next. So this approach has been well validated and published. With the biosynthetic approach, we are spiking these synthetic materials into highly characterized genomic background. We use a genome in the bottle, and we can actually change the background if you so desire. We've gone to show the patient-like performance, these mutation mix for sequencing and for pipeline assessment, or use as a full process control. We spike them into an artificial plasma. They can be highly multiplexed, anywhere from 10 to 50 different mutations per mix, and include all the SNVs, insertions, deletions, copy number gains, and of course we can include challenging and rare variants too. And there's a number of publications you can see here which have been peer-reviewed and shown to accept this technology for NGS assay validation. So here's more details about how we manufacture the product. We design them by creating a mix of clinically relevant variants. The variant is within a plasmid construct of about 800 base pairs with the variant in the middle. And you can see I've listed a number of clinically relevant variants here, from KRAS to EGFR, T790N, as well as 2 fusions, CD74 ROS1 and NCOA4 RET. Again, this is at the DNA level. So here's plasmid approaches. We also add to the same mix full genes, ERBB2 as an example, or meta example, where you're looking at copy number gains. And again, that then is mixed at the allelic fraction we need in genomic background. And we use the GM24385 wild type material. So we make the spiking, we quantitate by digital PCR, adjust as necessary for the application, and then we formulate and buffer. And most important for this application, we actually go through additional steps to make it patient-like for circulating cell-free DNA. And as shown here, we do the ctDNA, we do blending and fragmenting, and then formulate as either mutation mix or reference material in artificial plasma. And as shown here, the natural sized cell-free DNA, which is the material in gray, and the material in blue is exactly the manufacturing material. You can see how it has a very similar profile with a maximum peak around 170 base pairs compared to the natural material. This allows for good conversion efficiency as you make your library preps for NGS analysis. So again, we can customize this. We have off-the-shelf products, but most important is it can be adapted to whatever designs you need for assay validation, assay QC. Here's a list of the products we have in our somatic oncology portfolio. Much more detail on our website. But as I said earlier, we've moved into immuno-oncology with mutational burden and microsatellite instability, solid tumor, a number of types of products from tri-levels. It's either mutation mix, that's DNA in a buffer, or FFPE. Cell lines that have been formalin fixed and very well characterized. We have RNA fusions, the NTRK products, as well as some classic others like RET and MET, for example. And today we've been speaking about the ctDNA. And we also have heme disorders, myeloid, and lymphoma as well. So a really extensive group of products for NGS assay validation. I wanted to talk a little bit about the applications to give you some ideas on how to consider choosing reference materials and how they can be used in assay validation. This is a publication from our collaborators at Cancer ID, Ellen Heitzer. This was published last year in MDPI. And shown here on the left is results from an interlab study. This is a 1% variant allelic fraction multiplex mix of clinically relevant variants with SNVs, insertions, deletions. And we actually looked at the inter-run variability and the intra-run variability across digital PCR assays at different levels here at 2 nanograms, 4 nanograms, 8 nanograms, and then compare that to the GENER assay, the mass array, and then at some high throughput high genomic content Venio assay, QIAGEN assay, and SureSelect too. So we looked at multiple assays, and we're just looking at the CVs, and as you can see, the CVs reported here, inter-run. The expected is the background noise. That's the expected shot noise, kind of the background noise for these assays. So you can see relatively good, especially as you increase the mass on the digital assays. Your CVs get reduced down to close to 10 to 15, which is very good for these clinical laboratories. You can see the CVs are also very good for the mass array QIAGEN and the Venio assay as well. The other data shown in this paper is a different way of looking at technically evaluating assays. So again, here we're looking at input amount, and this is an inverse regression analysis, inverse binomial distribution. And we're looking at different inputs amount by digital PCR, again, 2 nanograms, 4 or 8. And then the mass array is right here. It's around 16 nanograms input. The NG assays are at 20 nanograms. And again, 72 nanograms with some digital assays. And you can see, for example, here at the digital assay at 4 nanograms, this is a 1% allelic fraction mix, but you can see the range at 99.9%. The range could be all the way as high as almost 2% down to as low as 0.45% variant allelic fraction with that type of distribution. So wider distribution, and the more mass you added, the narrower the distribution gets. This is a nice way to look at the different technologies for circulating tumor DNA using the same reference materials. And that's the point here. You can use the same reference materials. We have a good supply, and you really can compare different critical variables in validating these assays. Here's another study in collaboration with NIST, NCI, and a number of other groups. And here we're using a highly multiplex material. In this case, it had 40 mutations, and we're looking at a couple of other features, particularly extraction yield. One thing that you can use our materials for is choosing the appropriate extraction. So the circulating tumor DNA is in plasma. How do you extract it out? And here, a number of these laboratories looked at their methods that have been validated. And here we're showing the DNA yield as far as nanograms per mil by the laboratories at the different allelic fractions. Wild type, 0.125%, 0.5%, and 2%. So you get a nice view on the yield, but also the variability shown by the error bars at the different laboratories. So it's a nice way to assess the pre-analytical steps is using contrived materials. And the other thing we did is looking at the ability of these assays to detect different levels. Again, at wild type material, 0.25%, 0.5%, and 2%. And you can look at the range of you get for the different values, as well as the mediums are with the X's and right in the middle at the line cross at the X. So those are the medians and wide range for these plots. So again, a nice comparison of the results according to your assays. So much information can be gleaned from these multiplex materials as you think about validating your assays. Here's a paper published this past year that talks about another clinical application, and our materials were used to help validate the limits of detection. This study was published in Head and Neck Cancer. It's a clinical application combining ctDNA testing with HPV testing, both markers found in circulating plasma. The workflow is plasma, extract the DNA, library prep, and then look at both the ctDNA as well as the presence for HPV. The title is A Decline in Circulating Viral and Human Tumor Markers After Resection of Head and Neck Cancer. So our material was used to help determine the analytical sensitivity was determined to be at 0.3% across 23 different genes. So again, use of the contrived materials, in many ways following the blood pack recommendation to determine your limits of detection. Now, this method was used to monitor the ctDNA for these cancer genes in viral markers for these patients. And for monitoring, about half of the patients had detectable tumor markers in their plasma. And the technical limitations, which were identified with the idea to increase the clinical sensitivity above 52%, was that the assay was at this time only able to introduce 10 nanograms of DNA, and only some of these selected oncogenes could be tested. They'd like to expand the genes and expand the input DNA. And hopefully by making those types of changes, they can increase the clinical sensitivity. But I thought this was a fantastic example of where we're going, applying NGS testing to patient management. On mutational burden, let me spend a few moments talking about how we make these products and their application. So important for immuno-oncology companion diagnostics right now. So in this case, we're using tumor cells and normal cells from the same patient. We make mixes and then create paraffin blocks, and then we can make curls for your assay to be validated. We use whole exome sequencing. Here's the data for the tumor cell lines and normal cell lines. You can see reads, duplicate reads, and other QC metrics like on target, mean coverage, median coverage, etc. So these whole exome-based TMB scores were derived from this Agilent approach using Illumina NovaSeq and the FOCR harmonization project. So this has all been published in how we determine the mutational burden for these mixes. So we actually have 3 products that you can consider using. The early reports for immuno-oncology had a cutoff of around 10, score 10 as a cutoff using the Foundation Medicine. So we've actually bracketed that a little bit. We've got a score 7, a score 9, the mutations per megabase, score 13, 20, and 26 right now. So here are the TMB scores, just measuring the genomic DNA, and then going all the way to a tissue curl as an FFPE, the score as an FFPE. So you can see 7.2, 7.15, 9.5, 7.5, 12, 12, etc. So all this data is reported with our product. So how can it be used? We're at the point now we're still standardizing and harmonizing TMB assays. So our products have 100% tumor or 30% FFPE. And now we've moved to blood, looking at 0.5% tumor and 2%. So we have done some interlab studies looking at these materials against these NGS panels. Here's one study using a collaborative group working with us here at LGC from Illumina, the Broad, MSK, Neo Oncology, Medical College of Georgia, Semaphore, Roche, and Intermountain Health. You can see we took 4 of these scores, and these were looking at the genomic DNA, and we asked each laboratories to run it through their validated assays and report back the TMB scores. And you can see for score 7, the score 7.2, which is how we report it by whole exome, you can see the results from the different laboratories, some below, around 4, some above, some way above. But the importance was each of these laboratories can use this information and say, okay, if whole exome is giving you 7, and my lab is giving 5, what's the difference? Do I need to tweak my pipeline? So you just look across the data set and you can see there's differences, and actually closer results, the higher the score, but most importantly is to have analytical accuracy and precision down near the cutoff of these assays. So very valuable information as you think about validating TMB assays. So then we move to blood TMB, so important as well. It is emerging as a clinically valid selection marker for non-small cell lung cancer, for example. So blood TMB combines the challenges I just talked about for general TMB measurements, but also then you have more issues around pre-analytical availability since it's in blood. Perhaps since it is in blood, you have genomic DNA contamination, especially from clonal hematopoiesis, issues to deal with there. And then you need to establish your VAFs, very little fractions, and your TMB cutoffs once again. So lots of issues. Here's some interlab data using some R&D lots of blood TMB from our company in 2 different assays. So in this particular assay, we looked at 100% tumor, 0.5%, and 2% tumor. These again matched normals, and again this was just score 20. So you can see replicates at 0.5% or 2%. 0.5% is much lower at 11 mutations per megabase and 14 versus the tumor, solid tumor of 20. You can see at 2% slightly higher, and then you can see again the TMB 20 score over here for different replicates. Again, it gives the laboratory a sense of do they need to adjust their pipeline and how's their reproducibility. Here's another assay, again looked at 0.5% and 2%, but here we looked at 3 different scores, TMB score 7, 9, 26, and you can see the results. The black line here is the expected 7. You can see the results from the 0.5% less at 5.6, 2% higher at 7.7. TB score 9 came out much lower in this assay, 2 and 2.9, and TB 26 came up very close. Again, to me, this highlights we're in early stages of validating these types of assays for this application. Hence, you need to be able to use the contrived materials to help validate. Now, this is a report from the literature. I just want to highlight as we move from standard diagnostics of NGS for clinical oncology, moving to measurable residual disease by NGS. A couple points to make here. This is in product development right now, and the issue here is that we're looking at sequential and patient-specific disease monitoring. So a patient's been identified cancer, resection, follow those specific mutations for that person. It could be digital, could be a mass array, it could be an NGS assay. Solid tumor standardization, quality control, and validation protocols are needed. Now, we're actually stretching the bounds of sensitivity. So again, you do have to do standardization accordingly. The limits of detection can be an order of magnitude more sensitive than a standard ctDNA assay. For example, the FDA has approved a number of assays around 0.4% barium little fraction, and an order of magnitude less than that would be 0.04%. Real challenging. And the reference materials need to be able to assess the entire workflow, extraction, yield, look at these multiple variants. And I like this chart here from Anders Stolberg, Tony Godfrey, just published. And it looks at the numbers of ctDNA molecules based on DNA input. And this is basically one molecule is available to be detected at just over 3 nanograms of input. And then here, at this level of 0.01%. So we can imagine as we start talking about limits of detection at 0.04%, down in this range, the stochastic distribution or the distribution of those molecules in the sample is going to be a real challenge, something that you really need to consider as you push the sensitivity of these assays. So, but the field is moving forward with this, and I'm sure we'll have good approaches to monitoring patients of residual disease as we move forward. So our pipeline, we've got a lot of products in our pipeline. I've already talked to blood TMB, MRD monitoring, pan-cancer profiling, and cell-free RNA as well, looking at the expression patterns. We're moving into pharmacogenomics, host transcriptome analysis, and for cancer screening, DNA methylation is a hot topic, so we've actually got some products underway there. We're also looking at multi-parametric testing. So we do a lot of collaborations to advance this science and technology, and here's a list of partnerships and collaborations that we use over the years to help advance the development and the adoption of these reference materials. Wonderful group of investigators from the National Cancer Institute, Brown, Dartmouth-Hitchcock, NIST, Genosity, BloodPack I mentioned. We've joined the European Liquid Biopsy Society, Stanford Cancer ID, Forensic Cancer Research, particularly on TMB, and FNAH on ctDNA materials. And we actually have our own working groups. So I want to say thank you publicly to all these fantastic working groups. So when I close there, I say thank you. Here's my email. Please reach out to me with any questions you might have. Thank you for your attention.